Hi, hello, welcome to Physionic. If you're not familiar with who I am, my name's Nick. I'm a PhD candidate in molecular medicine. I work in a cell biology lab where we actually study the topic that I'm going to be discussing today, which is mitochondria. Not necessarily, well, actually, when I entered the lab with my metabolism background, I did end up doing a project that looked at nutrition, or really kind of the cellular state of nutrition. So I'm going to be presenting some information from a scientific review from the Journal of Cell Metabolism, which is one of the gold standards for research. And I'm going to be discussing essentially, or answering a few different questions. So how mitochondria respond, so cell energy production and changing their shape, to nutrition overload as well as starvation, which kind of synonymous with like fasting in this situation, as well as why mitochondria change their behavior. And I'm saying behavior, quote unquote. It's not like mitochondria think about these things. These are things that just happen on a biochemical level within the cells that cause changes. I'll be explaining what those biochemical things are that lead to this change in quote-unquote behavior from mitochondria. I just got done reading a grant for my lab. It took me about 4 or 5 hours to get through it. It's almost 20 pages long. And essentially what a grant is that it allows us to apply for more money to continue the work that we do. I think last year I released a video on a complex paper that we released in the Journal of Cell Biology, I want to say, or maybe it was JBC, I don't know, it doesn't matter, where we discovered a new form of autophagy when we knock out a particular protein that is involved in autophagy known as parkin. But now we're shifting our focus towards some other areas. I won't go into the details of exactly what, but I'm taking my metabolism background and kind of integrating it into my project. So I was kind of excited to hear some of the things that we're proposing, some of the areas that we've been going down, and I think my project is going to be really interesting. So hopefully one day when I do defend, hopefully I'll be able to kind of distill that or concentrate all of that information for you guys, so you guys can get some firsthand information from the research that I am specifically on beyond what I've already presented from last year. But without further ado, let's go ahead and jump into this video and we will break this down. So looking at how nutrition affects mitochondria, I will have probably 2 videos on this, because I'm going to have one, which is this one, explaining the mechanisms, what happens, et cetera, but then the second video is going to be discussing diabetes and obesity, or how gaining weight affects these processes, and some of the negatives and some of the compensatory mechanisms that our bodies, different tissues, cells within our tissues make. But I digress. So the question that we're examining, as I mentioned, is how do mitochondria respond to our nutrition? That's a really basic way of going about things. And it comes from this cell metabolism review, which I mentioned is kind of the gold standard, or one of the gold standards. I mean, Cell, the journal Cell, is right up there with Nature and potentially Science as well. So those 3 journals are kind of the top tier, and Cell Metabolism is kind of a sub-journal within Cell. It's also extremely highly rated. And this is a little bit older review. It's from about 7 years ago, 8 years ago. Mitochondrial dynamics in the regulation of nutrient utilization and energy expenditure. So I'm going to break all of this down for you. All right, so mitochondrial behavior, I'm going to discuss 2 different aspects of mitochondrial behavior. So one of them is the change, or the change in what's known as the morphology. So we've got our mitochondria in here, and what it can do is it can split apart, it can fragment, it can undergo what's known as fission. And if you've been following Physionic for a while, you've maybe seen me cover this before because, well, it's an important part of mitochondrial morphology. So this splitting apart leads to a single mitochondria and splitting into two mitochondria, and these tend to be a lot smaller. And I'll discuss a little bit of why that might be the case in this scenario, but there are a number of different reasons why this would occur. The second scenario is that you get two mitochondria that will fuse together and create an elongated or just a larger mitochondria. It doesn't have to be elongated, but what we're going to be looking at is elongated mitochondria, as you'll see. And I'll actually show you some real images of what this stuff looks like. So this is morphology changes in mitochondria. So behavior of mitochondria can change by the actual shape of the mitochondrion. Another way, however, and I've got this dividing line here, is that one scenario leads to a reduced level of ATP, which is adenosine triphosphate, which is cell energy production. And another scenario creates a substantial amount of cell energy. And we're going to figure out which scenario belongs to which, and then why there's this distinction between the two. Why is there this change in ATP generation? Why is the mitochondrial behavior changed in such a way that in one scenario you have increases in cell energy production and one you have decreases in cell energy production? So a lot of this is influenced, all within the context of what we're talking about, a lot of this is influenced by two things. So one is the supply of nutrients. So if that's blood sugar or sugar in general, carbohydrates or fats, so either one of those, and yes, amino acids also contribute, ketones contribute, all that stuff contributes. But I usually try to distill it down to the two major ones, which is carbohydrates and fats. So nutrients, and I'll explain this in a little more detail in just a second, but how much nutrient availability is there around the cell, then in the cell, and then that can then enter the mitochondria. And so those three scenarios. Outside of the cell, so in the bloodstream, in the interstitial fluid, then entering the cell, so it's not in the mitochondria, but it's in the cell. And then third is when it's actually in the mitochondria. And the second effector, the second component of this equation is not only the supply, but also the demand. So cell energy demand by literally every single process within the cell requires some level of energy. If that's ATP, it doesn't have to be ATP. It can be GTP as another example. And there are some other really tiny fractions that also belong to other areas as well. But ATP and GTP are the major ones. We're going to be focused on ATP because that is the major one. So cell energy in the form of ATP is then consumed by many, like thousands and thousands of enzymes within the cell. So if your ATP supply drops, then that means these enzymes can't function as well. Or if the ATP levels are elevated, then you do have enough ATP to allow these enzymes to function. So is the demand for energy by these cell processes also has an impact on mitochondrial morphology. Now, to get a little bit more into details with this, and I'll show you some more images of this, but nutrients enter the mitochondrion, and then there they undergo this process known as the TCA cycle. You may have heard of it as the Krebs cycle, but the TCA cycle, as I've got it labeled here, you don't need to know all these enzymes, but you have fats and sugars, carbohydrates, that then get converted to acetyl-CoA. Now, this molecule, acetyl-CoA, goes through this cycle, quote-unquote cycle, and as it's going through this cycle, what happens is you get the generation of new molecules. And the two major molecules, I guess I'll mention three major molecules that I want you to pay attention to, is with the introduction of fat, with the introduction of carbohydrates, in if for anybody that's interested, that knows about cell biology and metabolism, I'll say in an aerobic environment. If you don't know about that, don't worry about it for this at this moment. But these enter the mitochondria, and you get this generation of acetyl-CoA, the rotation through this TCA cycle. And as that happens, it produces these other molecules like NADH is one of them. FADH is the second one, and the third one, just to throw it in here, is also the generation of ATP. Technically, I believe you also get the generation of GTP, but they don't show it here. But for now, just know the big ones are NADH and FADH get formed when this acetyl-CoA goes through this cycle. All right, so there you go. There's an explanation of the supply and the demand and how the supply gets turned into these different intermediate molecules, and those will come back in just a second. So let's discuss now nutrition overload. How does nutrition overload, the overconsumption or the elevated amount of fats and glucose, etc., being present, how does that affect mitochondria? Well, let's discuss the morphology first. The morphology is what happens, and as I promised, that these are actual images of what happens. We've got our mitochondria, and what happens is that it fragments. So we get smaller mitochondria that end up being formed out of a single mitochondria. So it splits into one or maybe multiple other mitochondria. So, and you can see that in this image here. So this is before this fission occurs, this fragmentation occurs. And you can see that this is all fluorescent, so it's all fluorescent in green. So all the green is mitochondria, the blue is the nucleus. You don't have to worry about that. Just look at the green. And you'll notice that, if you kind of trace these with your eyes, you can see that they're considerably longer. They have some length to them at least. And this is kind of an intermediate state, as you'll notice in a little bit. But the point is that if you compare this image to this image, suddenly they're much rounder, they're much tinier, they're not as elongated as they are here. So does that mean that you have lost mitochondria? Not necessarily. All it means is that these mitochondria have changed their shape from a larger mitochondrion to a smaller mitochondrion. Okay, so to explain a little bit more on the energy aspects and why this might happen with fragmentation and whatnot, we have to look inside the mitochondria. So we're looking inside a single mitochondrion, as you can see up here on the left, and we're zooming into the inner membrane. So there's the outer membrane, then the inner membrane, and there's a space between the two membranes. This is called the intermembrane space. So we don't have the outer membrane shown here just because I didn't want to, I deleted a few different things because they just didn't add they just add more to look at and they just end up confusing. So just know that there's a membrane up here, which is also part of the mitochondrion, and it holds the rest of the cell away from the inside of the mitochondrion, which is here. Okay, so, and here you might recognize, yet again, this NADH and FADH. So those do come from the TCA cycle. And this is known as the electron transport chain. So these are proteins, and I promise there's a point to understanding this. So these proteins are integrated into the inner membrane of the mitochondrion. So with these electron transport chain proteins, what they do is they interact with this first one, complex 1. That's the name of this one, complex 1. It has a fancier name as well, but like oxidoreductase. But complex 1 for simplicity's sake, because this is already going to get too really into the woods. But complex 1 will then interact with NADH and essentially remove electrons. So that's where we get these two electrons right here. And those electrons get pushed or get moved from complex 1 to CoQ, and then will get delivered to complex 3, and then cytochrome C, and then complex 4. And the similar thing will happen with FADH. FADH will interact with complex 2 and generate more electrons, which will also go through this process. And ultimately, the end fate of these electrons is that they end up interacting with oxygens and hydrogens and creating H2O, so known as metabolic water. That's not that important. What is important, however, and what I will be pointing out, is that as these electrons are flowing from complex to complex to complex, going through until complex 4, where it's finally converted to water, there is a pumping of hydrogen, or protons, specifically protons, from the matrix, or the very inner section of the mitochondrion, to this intermembrane space. Now, why is that important? Because protons are positively charged. So what we want is, there's a negative charge. We want a membrane potential, so a negative charge inside the mitochondrion, and we want a more positive charge, or a more acidic environment in the intermembrane space. Here is where we keep most of the protons. So the mitochondrion keeps most of the protons. And then what happens, so you can see that the arrows are all pointing out of the matrix. And that's powered by these electrons that allow for these protons to be pumped back out of the matrix into the intermembrane space. Now, as this starts to increase with more and more protons, this protein right here, the ATP synthase, will then start rotating. And the way that it rotates is kind of like a mill. Have you ever seen a water kind of spill over a mill and the wheel starts to turn? That's exactly what's happening here. That the protons from this high gradient, the high amount of protons up here, will start to spill through this ATP synthase, rotating the ATP synthase. And in doing so, the consequence, the product, is the generation of ATP, or cellular energy. So in this direction, 3 times over, protons are moving out of the matrix of the inside of the mitochondria, the very inner parts of the mitochondrion. And in this situation, you have protons then moving back into. So it's a constant cycle going back and forth. So it has to be in kind of I don't want to call it equilibrium, but it has to be continuously moving back and forth, out, then in, continuously. So that's what would normally happen, and we get the generation of ATP as a result. So you have to have this balance of, do you have enough product, do you have enough supply, and do you have enough demand on both sides? So this is the demand, so the ATP would then be consumed by all the different cellular processes I was talking about earlier. And then for the supply, do you have enough NADH and FADH to actually run this cycle? All right, so now remember, we're talking about a nutrient overload situation. Now, if supply is so great and demand is not great enough, so demand is small and supply is high, then you have a high degree of NADH, you have high amounts of FADH, and they go through the same process, right? Donating electrons, it's going through this electron transport chain. But the problem is that because you're getting so much pumping of hydrogen out, and you're not getting enough of this drive to generate ATP, because you have enough ATP. You don't need more ATP. So you get a lot of protons that build up out here, but there's no drive through a metabolic demand right here through these reactions that would happen, enzymatic reactions and whatnot. So the protons are staying up here because there's only a small amount still coming through here to generate ATP relative to the amount of supply that there is. So as such, what happens is you have protons and electrons that start to leak. So instead of staying where they should be going, where they normally would be, for example, the electrons, instead of being pushed from one complex to another complex, they end up slipping, meaning that there's so much pressure building up, kind of, if you want to think about like chemical pressure, Think of it like a hose almost, that the water is coming in this way, and the opening is only so big, but the pressure keeps building on this side. Eventually, wherever there are weaknesses in these complexes, you're going to get slipping of these electrons. They're going to start bouncing all over the place instead of going to their respective spot, because the respective spot is taken by another electron, because there's no pulling, there's no release through the ATP synthase. So these electrons start to slip, kind of like a hose that has a weak point, and you start having a bit of water that starts poking out of the side, and then another leak coming out of another side. And it builds and builds and builds to the point where all these electrons are starting to freely be released from this electron transport chain, or not go to the electron transport chain, and it starts generating reactive oxygen species, which, if you've been following Physionic for a while, again, you know the different effects that ROS can have, ROS can have. However, there's a lot more that I can talk about ROS that I won't go into in this situation, but if you have an overabundant amount of ROS, then these molecules will then start damaging all the other molecules around them. And that is known as an overarching term as oxidative stress. So that is the situation when you have an oversupply of nutrition. The supply is really high. And this seems to lead to this fragmentation of mitochondria. And in certain tissues, there's something unique that happens. Now, one of those tissues would be, for example, brown fat cells. And I'm gonna tell you half the story now, and I'm gonna leave half the story for the next episode, but related to diabetes and related to obesity. But in a high nutrition overload situation, high fat, high glucose, high NADH production, high FADH production, instead of driving this electron transport chain, trying to push the ATP synthase to generate more ATP, even though there's no demand on the back end. So instead what happens is there's this, as I alluded to earlier, a leak in hydrogen, or protons specifically. So suddenly the mitochondria will start to generate more of these proteins that are essentially like pores. They're like circles that, well, I don't want to call them circles. It's not exactly right, but they're these pores that then integrate themselves into the inner membrane. And then from that point, now you have this unobstructed, this uninhibited release or movement of protons from the intermembrane space back into the matrix. And this is the reason why I say it's uninhibited, is that it's not coupled. That's a big key word, that it's not coupled to energy generation. So even though the cell may not need this added energy, it still produces this alternative pathway to speed this whole process up. Because at this point, the process is going very slow. There's a huge drive here to go. And here, then we get to these sections, and because this is kind of just moseying about, it doesn't have the drive by the cellular demand that I showed you earlier. Right here, these cell processes, it's not going to be moving very quickly. However, then the cell decides to introduce, not that the cell's thinking, but biochemically speaking, the cell then generates more UCP, or uncoupling protein, sticks it in here, and the protons can start bypassing the ATP synthase. This, in effect, reduces the amount of energy that is produced. However, it saves the mitochondrion from overproduction of reactive oxygen species. So it's a brilliant mechanism, and this is all according to this review. So some of this is certainly speculation. Some of this is definitely speculation. But based on the available evidence up to this point, that this then allows this to then speed up. So you're burning through these nutrients, you're burning through this NADH and FADH without the consequence of these reactive oxygen species being produced. As you're basing this sudden speed up based off of the proton gradient and not necessarily based off of the energy production needs that are necessary, because these energy production needs have not increased, but the proton gradient has been changed so that now you can smoothly go through this process. Hopefully that made some sense to you, that we're looking at it from two different perspectives. All right, so what about a low nutrition load? Based on what we already know and what we've seen with what happens to the morphology, the actual changes in shape, what would you expect to happen? Well, if you said the opposite, then you'd be right. And that's what seems to happen. So mitochondria will then elongate. And this isn't a great image, but I just kind of quick ripped it off the internet just to quick show you that here you've got kind of your intermediate mitochondria. You have some smaller ones. Again, this is through fluorescent microscopy. And the images aren't fantastic, but ultimately you see that it's more puncta based, meaning that you have more of these circular mitochondria. You have some of the kind of generally a little bit more tubular mitochondria for sure. So you have a nice mixture between the two. However, then when you go under a starvation media, or I guess a starvation scenario, and that can be something like fasting, or it can be something like just low nutrition, calorie deficit, etc. Of course, fasting is going to have an even greater effect. Then you start having these more tubular mitochondria where they end up fusing together to form more tubular, elongated mitochondria. So in this scenario, what happens is, let me quick throw myself back up here. In this scenario, what happens is that you don't have this significant drive through supply. You don't have a whole lot of supply to just burn through. It's not like the cells or the mitochondria are just trying to burn through their supply at this point, because they have a far fewer levels of this supply of molecules, fat molecules, glucose molecules, and therefore you have lower amounts of NADH and FADH, relatively speaking. So this is what's known as coupled mitochondria. So remember, with the uncoupling protein, it uncoupled mitochondria from energy generation. But now what happens, I was just checking if I had another slide to explain this, but now the demand for energy is higher than the supply. And in this scenario, now the mitochondria plays as kind of the middleman, or middle person, middle thing. And what it does is it starts to couple, it strongly couples the energy production to how it's going to then function. So as in, let me explain that a little bit more. So the demand is increased substantially, the supply is decreased. So as a result, it can't afford to build uncoupling protein and just waste this proton gradient through these electrons. It needs to use the electrons to the maximum efficiency. So therefore, the efficiency of mitochondria increase substantially. So as such, uncoupling protein does not get produced, and therefore we have this coupled mitochondria that simply bases its function off of the ATP generation. So in this scenario, unlike the other scenario we had decreases in ATP production because the mitochondria were just spinning or functioning based off of the proton gradient. In this scenario, it's not as much based on the proton gradient, although it's still important, but more so on the energy generation. So these enzymes that are not pictured need more ATP. Therefore, each electron ends up yielding, does not end up slipping from the electron transport chain, does not end up being used in production of ROS, but instead is efficiently transferred complex 1, complex 2, complex 3, 4, etc., therefore allowing for an efficient pumping of protons and therefore an efficient use of those protons then back into the matrix to generate ATP, to generate energy. Hopefully that made sense. And the final thing, the last thing, let me get rid of my image so you can see this more clearly. The last thing here is that not only, one of the problems that you might be thinking to yourself, maybe if you've gotten to this point, if you do, for example, fasting and you're really into autophagy and stuff, one of the issues is that, well, if mitochondria are becoming more efficient, let me cut to this real quick. If mitochondria are becoming more efficient, and with fasting we see increases in autophagy, which would presumably get rid of some mitochondria, which is known as mitophagy, why would that happen? Why would we have a need for mitochondria and yet we're getting rid of mitochondria? Well, this is called selective mitophagy, which I've actually had other content on as well. And it turns out that that is not what happens. In terms of, yes, autophagy may increase, but the mitochondria are protected from the autophagy. So here in a normal situation, under normal circumstances, let's say you've got a damaged mitochondria or whatever, it ends up being enveloped by the phagosome, and it will then be bound by what's known as a lysosome, and the autolysosome, I believe that's the correct term, autolysosome will then destroy this mitochondria. And then those components will be, some of them will then be recycled for the generation of other things within the cell. However, because of the elongation process, it's believed that this elongation process protects the mitochondria from autophagy. So normally, to undergo autophagy, you would want the mitochondria to be smaller so this phagosome can circle around it, enclose the mitochondrion, and then end up destroying that mitochondrion. However, in this scenario, it cannot happen because the mitochondria is far too large. So this is a really cool mechanism by which you can have fasting, but you can preserve your mitochondria, or at least the mitochondria that need to be used or that need to be preserved in that they are not damaged. All right, so in conclusion, mitochondria respond to, let me look at my notes here real quick. Mitochondria respond to nutrient availability. In a high nutrient availability scenario, mitochondria will fragment, reduce their energy production, and if possible, waste more energy, meaning that they don't go through that coupling cycle of generating more ATP, because they don't have that drive to generate more ATP. They end up wasting it by allowing the proton gradient to move through the uncoupling protein. When nutrient availability is low, mitochondrial efficiency increases as more energy is produced because it can't afford to use uncoupling protein to waste the proton gradient. Mitochondria will also lengthen and enlarge, protecting them from mitophagy, which is the autophagy of mitochondria. Now, in the next video that I'll be discussing is how does diabetes and being overweight affect mitochondria, or how do mitochondria affect these issues? So I've described some of the mechanisms in this video. However, ultimately, some of these mechanisms will end up failing in certain tissues, and those tissues will then be strongly driven towards a more diabetic state or a more obesogenic state. So if you're interested in that, then please click on the next video and I'll teach you all about that as well. And if you're not interested, then thanks for stopping by up to this point, and I'll catch you in the next one. Have a good one. See ya.